Well, good morning. Welcome to the 2024 Astronaut Scholar Technical Conference. We have so many scholars this year that we're breaking it up into 3 parts. So we'll have this morning, and then we'll return after the lunch and professional expo for the second part of the technical conference, and then we'll resume tomorrow as well. But good morning. For those of you who don't know me, I'm Caroline Schumacher, President and CEO of the Astronaut Scholarship Foundation, and we are very excited to have such a good turnout for our technical conference this morning, and hope that you learn a lot because these scholars have a lot to share with you. Couple housekeeping items with our scholars before I turn it over to our moderators. If you haven't checked in with the QR code, please do so. You don't have to get up right now, but later on, just go ahead and check in. If you want to sign up for headshots, see Janelle at some point, and she will get you signed up for those. And with that, I want to introduce our 2 moderators that have returned to our technical conference this year. I have Carol Fitzgerald Tyler, ASF mentor and trustee. And Ashley Monahan, 2018 Astronaut Scholar from Texas A&M. Okay, I was waiting for the... And with that, I'm going to turn it over to them so we can get this show started. Thank you so much.
Thank you, Carolyn. Good morning. Good morning. No, you got to do better than that. I've been up since 1:30. Good morning. Good morning. There we go. Yay. Well, hi, everybody. Thank you, Carolyn, for that wonderful introduction. It's a joy and pleasure and honor to be with all of you today. Carolyn mentioned my name's Carol Fitzgerald Tyler. I've been on the board of trustees for a few years. I don't really remember how many, but love this organization and absolutely in love with every single thing that you are all doing. You are changing the world, literally. So with that, I don't want to get us behind because then I get in trouble. So we're going to start with our first presenters. And the way this is going to work is we'll introduce our presenters. We'll give them a wonderful 5 minutes. We'll give them our full attention. And we'll learn so much together today. So again, delighted to be here. Delighted to see all of you. Delighted to see your families and your supporters. And let's rock it, shall we? We got the morning session. Let's go.
Okay, and then I get to cover the fun logistics. So for our kind of logistics today, each of you is going to get 5 minutes, okay? At 5 minutes, you're going to hear our alarm go off, and you can kind of wrap up what you're saying. But we really want to try and keep on schedule so that way the next session can also stay on time, right? So Will here has so gratefully offered to give y'all a 1 minute and 30-second warning. Okay, I know it can be really intimidating and you're kind of like zoned out when you're presenting, but try to keep an eye on him just so you kind of know how much time you have left. You will have a laser pointer that you can use, and you can advance your slides with the same pointer. Whenever you click to go forward on your slides, be sure to point it towards the back where our AV team is set up. That's where it connects to. Okay, so if you click it down here, it's probably not going to work for you. And so again, just keep an eye on the timer. This is supposed to be low stakes. Okay, you're surrounded by family, right? That's kind of the theme of the weekend. So take a deep breath. This is not supposed to be stressful. I know all of y'all are stressed out. I was too, but just breathe. This is supposed to be fun. Y'all are going to do amazing every year. This is probably one of my favorite aspects of this week, is getting to hear what all of you are working on. Because like Carol said, you're changing the world. So are y'all ready? Yeah. Okay. I'll accept it. We're going to get better over the course of the day. I have full faith.
All right. Thank you all. One other public service announcement for those of you who attended my professional development on how to do presentations a little bit better, et cetera. I'm here all day. I'll be in the professional expo. If you want some extra time with me, absolutely happy to do that. Also, I'm a great note taker. I'm going to take some notes on your presentation. If you want that feedback after we're all done today, I'm happy to work through that with you, and hopefully that'll help improve. I'm improving. I present all the time, and every day I go, how can I do better? So I'm happy to help you with that as well. So without further ado, let us start with the first person. Amanda Hobb, and she's going to talk to us about bioavailability and biocompatibility. Woohoo!
Good morning. Like they said.
Good morning. Good morning.
My name's Amanda. I study at Montana State University, and I did this research project with the Center for Biofilm Engineering. My project title is the bioavailability of leachate from 3D printed materials for spaceflight applications. And I'll walk through which each one of those bigger words means in context. So the context of my project is the WPA on the ISS. This really awesome assembly recycles like 99.9% of the water on the ISS. But it has a couple problems where biofouling is breaking filters. So that's one of the issues we need to solve before we can do long-term missions. There's a lot of different biofilm mitigation strategies. I've done different projects in each one of those, but my project this year is in monitoring systems. So building parts and sensors that can monitor the biofilm formation and levels within the WPA. So I'll walk you through a little bit of background on 3D printing. There are a couple different kinds of 3D printing. First is extrusion printing. That is a kind of printing where there's a coarser print because your aperture is based on the extrusion tip. Whereas resin printing can be much higher resolution because you're based on laser focus. But resin printing, though it's you can make tighter parts, you require gravity to print because you have a water basin or a resin basin that sits at the bottom. Finally, we have sealants, which I made with a mold press. So here's a couple of GIFs of what the differences look like between resin printing and extrusion printing. Extrusion printing prints right side up. Resin printing prints upside down. So these are my materials that I printed. I had my machined controls were glass and polycarb, and then I had two different kinds of PLAs. I had a surgical guide, and then my silicone as my sealant. So my experimental design, I started with allowing my coupons, which are those little circles of material, to sit in sterile water for 2 weeks, measuring the carbon, the dissolved organic carbon that came off of them. Then I used that water, which is then called leachate, in my second experiment, where I diluted super concentrated minimal salt media, which basically gives microbes everything they need to live, but not everything they need to grow, so that the carbon that they grow from is either residual carbon they've stored or coming from the leachate. And then I measured growth and the organic carbon that came off of that. So my abiotic results, you can see that leaching happens for every 3D printed material, which is kind of interesting. Most significantly for eSun and surgical guide, which are resin printed. But what's super interesting is then when I tested this in biotic in triplicate with many, many biological replicates, all those colors are kind of blending together. There's not a significant difference between control and the tested leachates. So then I decided, because we know that there's problems happening when we put these into our systems, they're biofouling. So then we decided to take some pictures. This is like the control picture. So this is a really thin monolayer biofilm on glass, which is one of the controls. This is of Rostonia. I missed back there, my two bugs I used is E. coli, which is a really common laboratory strain, and then Rostonia insidiosa, which is off the ISS. So this is pictures of Rostonia. And you can see that on the different materials, we get super interesting different biofilms. So this is kind of where the future is leaning. My conclusions from this section of the project is that all the 3D printed materials leach carbon. And they reach steady state at about 1 week, which could be interesting for pre-processing before you put them in the system. But that carbon does not have significant impact on growth when in leachate. My future work, I want to chemically identify what's coming off of these products. I want to test all my materials in synthetic wastewater rather than just sterilized water, and continue to test biofilm formation through spectroscopy and look at the different surfaces and how they're interacting with the microbes. And these are my sponsors. I'll take any questions you have.
Who has questions? We have a couple minutes to take some. Anybody have a question? Great.
What did the two false colors in your microscopy images represent, the red and the green?
It's dead live stain. So green is alive, red is dead.
Just calcine and FD?
Yeah, and actually what was super interesting, oh, I don't have my slides anymore, but there was some background fluorescence from the different materials, like the surgical resin fluoresced. So we want to take more images to make sure what we're picking up on that is actually, let me see, go real fast. Make sure what we're picking up on that is actually the bugs and not just background imaging. So definitely need to do more replicates on that. Any other questions?
One more question. Who has another question?
Do you think the differences in these biofilms comes more from the structural properties of the like little the coupons or like the chemical properties of them?
Yeah, I totally think it's a combination. I think the hydrophobicity and like that surface contact angle is super important for biofilm, especially at the beginning, initiating stages. So I want to measure that surface contact angle. But I do think because we see, even though these were all inoculated with the same amount of inoculum and they had the same amount of growth material, we can see that different biofilms, like different thicknesses form. So I do think there's some consumption going on, but definitely need more images and data to support that. Okay. Thank you.
Thank you, Amanda. Our next presenter, Nathaniel Carey, is going to be coming to us remotely, and he's going to be talking to us about 3D extrusion bioprinting of engineered human myocardium. Nathaniel, are you with us?
Yes, I am. Can you hear me? Can you see me?
If you could speak up a little bit louder, that would be great.
Just a
Perfect. Thank you so much.
Can you hear me now?
We can hear you now.
Perfect. Thank you. Alrighty. So my name is Nathaniel Carey, and I'm here to tell you about my summer research project entitled 3D Extrusion Bioprinting of Engineered Human Myocardium. This was a summer project I conducted with the Max Planck School Matter to Life and the University Medical Center in Göttingen, Germany. Starting off with some background on what is engineered human myocardium, or EHM. It's currently being manufactured in Göttingen in two forms. On the left, we can see small rings, and on the right, what I was working on were patches, which range in diameter from about 1 centimeter up to around 6 centimeters. These patches are designed to be surgically implanted onto large areas of necrosis left over on the heart after severe heart attack, or myocardial infarction. That's MI. Now, currently the patches are made by a molding process. And the issue with the patches, when you pull them out of the mold, is the heart muscle cells are arranged in many different directions. So we don't see the consistent linear polarity, which is characteristic of healthy heart muscle tissue. This results in severely reduced structural integrity, as well as weakness and poor synchronization of the contraction of the heart muscle patch. To tackle this issue, we take inspiration from 3D bioprinting, and specifically from a 2013 paper out of the University of Tokyo, in which roughly meter-long filaments of synthetic heart muscle fiber were printed. I like to think of these extrusions essentially as a churro. The middle of the churro is made of a mix of collagen, fibroblasts, and cardiomyocytes. That's the same exact mix that we use in the heart muscle patch. But they're surrounded by a shell of alginate, which is then stiffened by calcium. Now, we would love to directly print these fibers from a standard 3D bioprinting platform, but in previous work, we've seen that the 90-degree extrusion angle, which is characteristic of typical bioprinters, is very rough on the outcoming muscle fiber, and it results in significantly reduced cell viability. To tackle that problem, we've decided to shift the entire bioprinting platform to a 6-axis Denso robot. And this would allow us to significantly increase the extrusion angle labeled alpha in the top right to a much shallower angle, while still being able to weave complicated structures into larger forms of heart muscle tissue and maintaining proper cell viability during this process. This represents multiple significant technical challenges, chief among them being optimizing our extrusion path that is done through Robot Operating System, or ROS, as well as communicating between the Denso robot and the system of Arduinos, which controls the bioprinter head. Once we've optimized these components into being able to print our woven heart muscle patches, we still have some adjustments to make regarding the material that surrounds the outcoming muscle fiber as it leaves the bioprinter head. That material is called the print bed, as well as the material from which the churro itself is made, the extrusion medium. And really what we're looking for is always how do we get the best biocompatibility, as well as specific to the extrusion medium, how do we select materials which facilitate the migration of the cardiomyocytes through this matrix as the heart muscle fiber matures? Again, after we have our fully printed heart muscle patches, we will need to improve our imaging techniques. So in the bottom left, we can see an example of a patch which was made via the molding process, then exposed to a clearing solution. This makes it extremely transparent. It's really a cool process. But in this process, we actually damage the patch quite a bit. And this won't be suitable for analysis in the future. Coming up on the horizon, we will need to develop ways to measure the strength of contraction of the entire patch, as well as an overall biocompatibility assessment before the really exciting part, which is clinical trials in real human patients. Thank you for listening. Do we have any questions?
Thank you very much. Do we have a question for Nate? Anybody? Anybody? All right. No questions, Nate. You did a great job. Thank you so much for joining us.
Thank you.
All right. Our next presenter is Dilshan Rajan, and he's going to talk about auditory cortex of guinea pigs. I love it. Welcome.
Hello everyone, my name is Dilshan Rajan, and I'm going to be presenting about the effect of paired stimulation on the brain's perception of sound. So the brain is constantly adapting and changing, which is a concept known as neuroplasticity. Using this concept of neuroplasticity, my lab wanted to see how we could affect the auditory neurons to change how the brain perceives sound. With that information, we could develop treatment methods for tinnitus, which is a phantom ear ringing and a form of chronic pain affecting over 10% of the world's population, with very few current treatment options. To induce neuroplasticity, we use the concept known as paired stimulation, which is the combination of two different stimuli together. In this case, we're using sound stimulation and electrical stimulation in combination, which causes an increased release in neurotransmitters, which help facilitate neuroplasticity. So the main purpose of this experiment was to identify how the brain's response to sound can change using the pairing of sound with electrical stimulation to treat tinnitus and hearing loss. So our experiment was done in 3 guinea pigs. The right primary auditory cortex, which is right here in a human, was exposed, and a 4-shank 32-site recording electrode was inserted to record the neural activity. An ear bar and speaker were then inserted into the left ear, and a stimulating electrode was also inserted into the left ear for the electrical stimulation. The stimulation consisted of 1,000 milliseconds of 1-octave narrowband noise and 1,000 milliseconds of 15 hertz electrical stimulation. Before and after paired stimulation, we recorded the behavior of neurons in the cortex, and the main metric we measured was neural spiking. So this graph is an example of a single neuron's response to a specific sound. The x-axis is time in milliseconds, and the y-axis is the spike amplitude. Each of the red dots at the bottom represents a notable spike or action potential, which is the neuron's reaction to that sound being played. This is done for several different sounds and frequencies, but I've only shown one here because it's a lot easier to understand. Note how the neuron is dormant before and after the sound is played, and there's a huge spike in activity immediately after the sound. Now, if we take all of the neural responses for a specific sound and find the time window where the highest firing rate occurs, we can find the firing rate response for the given frequency to create this plot. This plot shows the response of all the neurons to a specific sound frequency, with the x-axis showing the sound level or volume in decibels, and the y-axis showing the firing rate of all the recorded neurons to that sound. The blue line shows the response before paired stimulation, and the red line shows the response after paired stimulation. These plots are also made for several other frequencies that aren't shown here. And this particular graph shows that the brain's response to this given frequency has increased due to the paired stimulation, and that increase is more noticeable as volume increases. So the main results of this preliminary data shows that pairing sound at certain frequencies with electrical stimulation makes the brain more sensitive to that particular frequency. So using this concept, we can develop treatments for tinnitus by using paired stimulation to either reduce the brain's attentiveness to tinnitus frequencies or to amplify all the other frequencies to drown out tinnitus frequencies. And in future experiments, we plan to test different parameters to better understand what types of changes are caused and how we can better control those. And the figure here kind of shows a future vision for a medical device that could be taken home that patients could use for tinnitus that uses the concept of paired stimulation. Thank you.
Thank you so much. Any questions? There's right behind you.
Thank you. Did your group account for the possibility of habituation to sound without the electrical stimulation, like just getting used and more responsive to that sound?
Yeah, so there's a good time delay in between to test for that. And before we do the paired stimulation, we'll test that, and then we have ways of measuring the adaptation over time, but it usually doesn't last long enough for it to make a significant effect.
One more question.
Why'd you pick the model that you did for animal?
Yeah, so the guinea pig's entire auditory cortex is actually mapped out. So when we insert the electrode, we're able to identify what layers of the cortex we're in, and basically we can understand exactly where we are in the cortex, so we can kind of compare our data to what's currently out there as well.
Thank you so much. Very interesting. Thank you. So now let's welcome Catherine Keene, and she's going to talk about determining genetic control of recombination. So welcome, Catherine.
My name is Catherine, or Katie Keene, and I'm from Tufts University, and my project is looking at unraveling the mechanisms of length variation in the RS3 repeat of the AVPR1A gene in Saccharomyces cerevisiae, which is budding yeast. So I'm going to start by talking about the importance of repetitive DNA, because that is what my lab investigates. Repetitive DNA sequences make up about 50% of the genome, and they are largely not harmful. However, there are a lot of these regions are associated with irregular structures or molecular process inhibition, and they're very relevant to many diseases. For example, Huntington's disease is a repeat expansion disorder, so the repetitive DNA creates issues with the protein, which causes it to become non-functional. For example, imagine if you try to say the same word over and over again, you're gonna be more likely to make mistakes. It's the same thing with your proteins. They are gonna get confused if they read the same sequence over and over again and are more likely to make mistakes. So So a person with Huntington's will start with this length of repeat earlier in their life, but as their life goes on, it'll get bigger and bigger, and eventually the protein will be non-functional. Also, repetitive sequences are more likely to form these types of structures, which can make molecular processes very difficult to carry out. So my focus is on the AVPR1A gene, which is a receptor for vasopressin, And AVP's main function in the body is to regulate urine concentration. You also probably know it by its other name, antidiuretic hormone. It also plays a significant role in social, sexual, and mating behaviors. And there's this really unique relationship in voles. So they've actually one-to-one correlated. In voles, these voles are very similar. The prairie vole is monogamous though, and the meadow vole is promiscuous. And they've actually one-to-one correlated the presence of this microsatellite region, which is repetitive DNA. The meadow voles don't have this repetitive DNA, and the prairie voles have this repetitive DNA. And the prairie voles are monogamous, and the meadow voles are promiscuous. And it's just because of that lack of or presence of the repetitive DNA. So in humans, we have a very similar AVPR1A gene to voles. And one of the repeats in the human gene is called RS3, and it's right here. And in the human population, there's a ton of variability in this repeat, and they haven't been able to correlate one-to-one promiscuity versus monogamy in humans with this microsatellite region, but they have found some interesting relationships. For example, there are divergences in protein expression and brain physiology, and also behavior. I think they found that people with longer RS3 alleles are 50% more likely, or 50% of people who had longer RS3 alleles were never divorced or something. And there are more studies like that are really interesting. So there have been a lot of studies looking at the social implications of the repeat length, but we don't know how the variability is generated, and my lab is not a social behavior lab. My lab is a molecular genetics lab, so we're looking at how these molecular processes cause the variability to exist. And we use yeast as a model organism, and this is because it's very simple to use. You can really easily take out certain genes and also insert genes. So for example, we insert this bit of DNA, and it probably does not make very much sense to a lot of people, but basically what we try to do with this is we want to select for a certain condition. So in this case, we're trying to select for repeats that change length. And basically what happens is if the repeat changes length, the yeast will be able to grow on a certain media, whereas the yeast that didn't have length changes can't grow in that media. So in that way, we can calculate the rate of the length changes. And using this system, we were able to get the entire variability of the RS3 repeat that's seen in humans using the yeast system. We're also able to determine the broad molecular process, which is responsible for the changes in the RS3 repeat. And now we have to do this thing called molecular. It's like a very classic molecular genetics assay where I don't know what happened with the spacing, but what you're supposed to do, or what you do, is you select a gene that you think is involved in the pathway, you take it out, and then you measure the rate of your selectable event, and then you compare that to the rate of the strain that still has that gene. And so basically, these are all proteins that we've knocked out and determined the rates for. Each dot is like an individual test that had like 12 plates in it. So the blue ones are, the blue ones are inhibitory, the red ones are involved because the rates decrease, and the gray ones are not involved. And so we just do that a ton of times, and we look at everything. And so the conclusions are basically, that was a big chart, and no one probably knows what it means, but that's just kind of the scale of what we're doing. And what the results indicate broadly is that the process is regulated by a non-canonical DNA repair pathway, which means it's an atypical DNA repair pathway, because basically proteins that we thought would be involved are not involved, and proteins that are involved, we're kind of surprised that they're involved. So we're still working to figure out what's going on. And
thank you so much. So we have time for one question for Katie. One question. Anybody? Well, I never woke up this morning thinking I was going to hear the social implication of promiscuous bowls. But thank you so much, Katie. Very interesting.
My PI always says that before you start dating anyone, you should find out how long their RS3 allele is.
Or do they have a vole? Thank you so much. Next, let's welcome Cecilia Meinking, and I love her title, What's in the Water? Welcome, Cecilia.
Hi, everyone. My name is Cecilia Meinking. I'm currently a senior at Miami University, which is not in Florida, it's in Ohio. And today I will be talking about my research on the development of single-stranded DNA aptasensors for the detection of per- and polyfluoroalkyl substances, or PFAS compounds. So first I need to get into what are PFAS compounds and why are they important to study. They were initially developed during the Manhattan Project, and they describe very chemically stable compounds. They are involved in many different manufactured compounds that we associate with our daily lives, including things that have non-stick coatings or waterproof coatings. The thing is, extensive manufacturing use and a lot of waste dumping has increased PFAS concentrations very much in where regions of manufacturing are very heavy, like the East Coast and the Midwest. And this wouldn't be a problem, except it is, because PFAS have been associated with many illnesses in both adults and in developing children. So adults are seeing a much greater increase in cancers, and developing children are seeing most oftentimes lower birth weights and reduced vaccine responses. So the best way to prevent people from developing illnesses is to prevent them from being exposed in the first place. So we need to do a lot of frequent testing. And the current gold standard for testing is the involvement of liquid chromatography and mass spectrometry. And this method is very good at detecting for PFAS. However, it has a lot of limitations, mostly including its price. This instrument alone can cost upwards of $400,000, along with the necessity of a centralized laboratory, the need for highly trained personnel to operate it, and other expensive instrument maintenance, which can limit the amount of testing that we can do. So the centralized goal of my project is to utilize functional nucleic acid, or FNA technology, to develop a more portable PFAS sensor for on-site detection. So what are FNAs? They are very readily programmable pieces of DNA that can do basically any task we want for it. So you can have DNA enzymes, which are catalytic in nature. Their job is basically to do the catalysis of a certain chemical reaction that usually produces a signal that we can measure. Then you can have aptamers, and their job is to bind to either a specific chemical or very similar class of molecules. So in this case, it would be PFAS. And then both of these types of FNAs can be amplified and selected for using a method known as SELEX. So my job, and with this research project, I was wondering, can we develop a sensor array to improve selectivity for PFAS using FNAs, and can aptazymes, hold on to that, improve sensitivity for detecting PFAS? But I mentioned aptazymes. I didn't talk about that before. What are those? It is the combination of both an aptamer sequence and a DNA enzyme sequence to make a single-stranded DNA molecule that can do the function of both of those structures. So you can have the ligand binding portion of the aptamer, it triggers a conformational shift to the DNA enzyme, and then you can get your chemical reaction that you can measure. So this product from that chemical reaction is blue in color, and we can measure that through absorbance. So I have basically 4 different aptazyme designs, the first set of which does not include a portion of the aptamer that would be here, known as a hairpin. We were a little worried about selectivity with that. And then the second round of designs does include that hairpin construct, and it also includes what's called a corresponding blocker sequence that's just corresponding in the DNA. It will bind to it and then detach when PFAS binds. And then the other thing that I did was I split up the DNA enzyme sequence asynchronously. So there's usually 1 or 3 groups on one side, and then you can flip it for the other, because 5' to 3' directionality in DNA. So what I noticed for the hairpin excluding structures was that there was a really interesting response for PFOS, which is perfluorooctane sulfonic acid, which is interesting because I went back, never mind. I have PFOA in this figure because the aptamer sequence that we're using was designed for PFOA specifically. So it's interesting that we're seeing an increased response to PFOS in both of these structures, which may have to do with something in an aptazyme structure. There's something related to steric hindrances. And then for the hairpin-containing structures, we see a really similar thing where there's this weird jump up with PFOS versus PFOA. And it's like, oh, that's interesting, because it's not supposed to do that. It's supposed to be more responsive for PFOA because that's what the aptamer was designed to do. But maybe something in an aptazyme construct makes it more selective for PFOS instead, which could potentially allow us to do an aptamer or aptazyme array. So to conclude, we saw that all of the app design constructs that I designed had increasing signal activity with an increasing concentration of PFAS, which does show, okay, this system is working. We can then optimize it to develop a type of array. And we also saw both the increase with PFOS and a linearized relationship for PFOA in 3 of these 4 construct designs, which means that, oh, we could potentially develop a calibration for that as well. For the future, I'd like to analyze different app designs, including a different DNA enzyme than what I used. I currently use one called PW17. I would like to use one called B730. Does the exact same reaction. However, B7 had a little bit of a different activity, kind of a more robust Vmax in preliminary studies, so we'd like to look at that. I also would like to characterize a library of sensors for PFAS and potentially develop a pattern-based detection method, and then test environmental samples using that array. Lastly, I would like to thank everyone in the Yale Lab at Miami University and the Arnold and Mabel Beckman Foundation for funding my project. I will now take questions.
Oh, thank you so much. Thank you. Well done. Questions? Go ahead. It's a race. Who gets there first? There we go. Yay.
I have a very basic understanding of PFAS, but one of the characteristics that I have been led to believe, and correct me if I'm wrong, is that they're relatively inert. And I was just wondering if you could describe the mechanism in which your app designs are able to interact with them further.
So to my understanding, they're able to do binding based on the number of fluoroalkyl groups. This data I couldn't show, I was really sad about it. But when I looked at aptamer characteristics individually, we saw a really interesting trend in which there was a sweet spot where there were about 6 to 7 fluoroalkyl groups where it had the maximum amount of activity, whereas if you went lower or higher than that, it had minimal activity. So I'm wondering, potentially something sterically with the size and number of fluoroalkyl groups could be why this aptamer, specifically as its own component, behaves how it does, whereas there's another part of my project using CLEX to design different aptamers as well, where we're trying to figure out, okay, based on the number of fluoroalkyl groups, can we get different aptamer sequences that will bind differently based on that number? Because that's what we're noticing. It's not necessarily functional group, because different PFAS have different functional groups, but I'm thinking it has to do with number of fluoroalkyl groups, to my understanding.
One more question. Anybody? No? Well, thank you so much, Cecilia.
Right,
it's good to know what's in the water. Next, we'd like to welcome Shelby Sliger, and she's going to talk about characterizing the role of H2A.Z in homeostasis. Welcome, Shelby.
Okay, hi everyone. My name is Shelby Sliger, and I'm a biochemistry student at Purdue University, and I'm going to be talking to you today about characterizing the role of H2AZ in plant development and heat-responsive gene expression. So a little bit of background. I use the model organism Arabidopsis thaliana, and it is a flowering weed depicted on the left there, and it is actually the first plant to ever have been grown in space in 1982. I specifically study chromatin, which is a complex of DNA and histone proteins that exists in all of our cells. And it's actually the fundamental chromatin subunit is called a nucleosome, and it is made up of 8 of those histone proteins. So why study chromatin in Arabidopsis? So histone modifications can alter the gene expression, which drastically affects plant growth and development, which is important in understanding agriculture. And H2AZ and H3K27 trimethylation are 2 of those histone modifications that we know affect development and are also conserved across many plant and animal species. So H2AZ is commonly enriched in the body of many environmentally responsive genes, and it actually swaps in for H2A in the canonical nucleosome right here, and it's encoded by 3 genes called HGA8, 9, and 11. And then H3K27 trimethylation, or histone 3 lysine 27 methylation, is actually a repressive epigenetic mark, meaning it tells genes to shut off, and it's commonly enriched in the body of genes associated with differentiation and development. Okay, so we did a previous study where we noticed that of the many genes, about 20,000 or so that exist in Arabidopsis, H2AZ and H3K27 trimethylation are often co-localized, meaning that they appear together at the transcription start site as well as the gene body. And so then this led me to my hypothesis that the loss of H2AZ might actually be sufficient to alter H3K27 trimethylation in plants, meaning H3K27 trimethylation is dependent upon H2AZ. So how we conducted this study is we took 17-day-old plants, wild-type plants with no known mutations, and HG9, H11 plants, which are missing 2 of those 3 genes that are important to encode H2AZ in plants. And I conducted RNA sequencing, which is used to essentially examine the total gene expression, and chip sequencing, which is used to assess the chromatin modification state across the gene body. So these are a snapshot of my chip sequencing results. So we learned that wild type and HG9, H11 actually have nearly indistinguishable patterns across the gene body for H2AZ enrichment, starting at the transcription start site and ending at the transcription termination site for protein coding genes. And this is extremely surprising to us because it's missing 2 of the 3 isoforms of H2AZ. So that tells us that the remaining isoform is actually sufficient for seemingly most functions. However, you'll see here for the H3K27 trimethylation graph, we still see a significant decrease in the amount of this histone modification associated with differentiation and development. And this also is consistent with the phenotype of the plants because they flower early and also have a different rosette size than normal wild-type plants. So while this initial discovery was really exciting, I was interested in finding out if this also had an effect on the environmental responsivity, which I mentioned earlier was an H2AZ-dependent function. So I conducted a study using qRT-PCR, and I selected 7 different genes here that are all heat responsive or drought responsive. And I took my plants and grew them in a plate incubator at 17 degrees Celsius. And then I shifted some of them to a hotter temperature at 27 degrees for either 15 minutes or 4 hours. And so what I found here, focusing on that blue and yellow bar for wild type and HG9, HG11 mutant plants respectively, that there is a significant decrease overall in the induction of these important heat responsive genes, which suggests to us that the HG9, H11 plants are less sensitive to heat despite having the same amount of H2AZ overall. And so our conclusion here is that the type of H2AZ, rather than just the amount, has a significant impact on plant development and environmental response. And so that allows us to ask important questions like, how might crops utilize H2AZ to respond to environment? And how might H2AZ and H3K27 trimethylation inform uncontrolled differentiation and development in cancer and animals in general? And I just want to acknowledge my lab and ASF for letting me present here today, and I'm happy to take any questions.
Thank you so much. Questions for Shelby?
Why did you choose to knock out 2 genes at once? And did you determine that beforehand? And have you determined if there are interactions between them?
Yeah, that's a really good question. So when you knock out the protein that deposits H2AZ, so it would be an H2AZ null, the plants basically don't grow at all. So they will flower super early and they don't produce any seed, they're sterile. So we knew we wanted to look at H2AZ deficient mutants, but ones that would still grow, right? So we were under the understanding that they would be H2AZ deficient, but actually we made the discovery that they really are not, which allowed us to test an entirely different hypothesis altogether.
One more question. Anybody?
How do you think that H2AZ is actually changing H3K27 methylation? How is a histone protein changing the actual methylation of another histone?
Yeah, that's a great question. So the protein that catalyzes H3K27 trimethylation is called PRC2 in plants. It's called EZH2 in animals. And so we think that enzyme might actually prefer the H2AZ enriched nucleosomes as its substrate, and that's a hypothesis we're testing in the lab right now.
Cool, great work.
Thank you, Shelby. Thank you very much. Next, we would like to welcome Lily Tam, and she's going to talk with us about building bridged nucleosomes and truncated proteins. Welcome, Lily.
Good morning, everyone. My project was performed at the University of Rochester in the Department of Biochemistry, and I will be talking about building bridged nucleosomes and truncated proteins to assess linker histone folding. So, chromatin is the unit composed of DNA, and it gets wrapped around these histone proteins, which you learned about in the past presentation. But how does chromatin go from this very loose beads on a string configuration all the way into chromosomes, which separate during mitosis? The secret is linker histones. So, linker histones are a protein whose expression varies across different species. There are many subtypes expressed in different tissues. In humans, there are about 5. And linker histones bind to the nucleosome, and then the C-terminal domain condenses and collapses, wrapping around these linker DNA arms to pinch, forming the first layer of chromatin condensation. Some key terms of this presentation will be histone, nucleosome, linker DNA, and steric hindrance. And there are 2 different potential models reported in the literature, a dyad binding and an off-dyad binding. Off-dyad, the CTD would only grab one of the linker arms, so we're not really sure how that would play into condensation. So I started off building truncated H1 mutants. H1 has 3 domains, the lysine-rich intrinsically disordered C-terminal domain, a central globular domain, and a short N-terminal domain. So we have 3 mutants, NTD and GD, GD and CTD, and just GD alone. And our hypothesis is that these truncated H1 proteins can reveal which domains promote condensation upon binding histones, and the readout that we used is fluorescence resonance energy transfer, also known as FRET. So with FRET, we excite one of the probes at a certain wavelength, and this prompts an emission. And based on the proximity to the acceptor probe, the acceptor is also going to fluoresce. And this tells us how far they are away from each other. So if we have the CTD binding and then collapsing, we expect to see the probes getting closer together, informing us of whether collapse has occurred. And so far, these results have not been published, but we are seeing the importance of the CTD to promote histone collapse. And we're interested in seeing how this varies across different subtypes, which might inform the layers of expression that you see in different tissues, especially in genetic diseases when you don't get the expression of a certain protein. My next project involves building bridge nucleosomes. So we have our histone, and we have, this time, the probes on the linker histone itself. And the idea is that steric hindrance can affect nucleosome folding. So our lab has previously shown that nucleosomes alone will condense more rapidly than in an array, like there's some steric hindrance from the other ones being present. So we're trying to model this by building a bridge nucleosome. So, the bridge is here, and that would create steric hindrance that might prevent the CTD from collapsing. And we want to create a bridge of varying length, like 20 to 70 base pairs, so we can stimulate different states of nucleosome steric hindrance and see how that affects the collapse. Over the course of this project, we've discovered some interesting things about ligating PCR DNA. It's harder than expected. So we use tailed PCR to create the base of this DNA that wraps around. And then the idea is to put on duplexes, which will be complementary to the bridge and allow us to put in bridges of different sizes on the same construct. And we've been able to get either of the duplexes, but not both at the same time so far. This is what the majority of my products look like. We can see here in lane 2, depletion of this 167 base pair construct, so one of them is binding, but the other one's not. And we're trying to work out how to get both of them to bind in order to get the bridge in and actually make the project work. So some future directions are finishing to build the bridge nucleosomes and then combining the projects. If we put a truncated mutant into this and we have the probes on the linker histone, can we see if CTD is the only part affected by steric hindrance, and are the trends that will emerge uniform across different H1 subtypes? I would like to thank my PI, Jeff Hayes, great guy, as well as my mentor, Ashok Kumar, who is now a research assistant professor in the department, the Schwartz Scholars Award at the University of Rochester for funding my research full-time last summer, and the Astronaut Scholarship Foundation for allowing me to present today. Here are some references.
So who has a question for Lily? Anybody?
I'll ask one. So I'm curious, the truncated mutants that you're using, are there any human individuals with those types of mutations? Are those people even able to live?
That's a really interesting question, and we have not seen any reports in the literature so far. This is also very basic science. It's really tough to even get to image what's going on at the level of the nucleosome. So if there are, for one, it would be really interesting to see if the expression of other variants in the same tissue could compensate for that lack, maybe it just gets expressed and doesn't get used. But that's something that maybe we'll be able to get clinical reports on someday, but probably not right now.
Any more questions for Lily? No? Thank you so much. Fascinating. Next, let's please welcome Chloe Neihart, and she's developing a minimally invasive method for assessing individuals' chronotypes using hair follicle cells. Cool.
Thank you. Hello, everyone. My name is Chloe, and today I'm going to be discussing my current undergraduate research project. I've been exploring a novel approach to assessing the human chronotype using hair follicle cells. So the human chronotype is the individual's tendency to sleep and wake. It is a behavioral manifestation of the circadian rhythm. The circadian rhythm is shown in this clock diagram here. It is the periodic changes a person experiences to regulate internal processes. So for example, a person may experience their highest blood pressure at 6:30 p.m. or their lowest body temperature at 4:30 a.m. Insight into an individual's chronotype can be used with chronopharmaceuticals. This is basically applying the individual's chronotype to enhance drug delivery effectiveness. And in order to use chronopharmaceuticals, we need to have an understanding of an individual's molecular footprint of their internal circadian clock. So these are the genes that are oscillating to regulate circadian rhythm. So my project is developing a minimally invasive and accurate method of assessing the internal circadian clock and the human chronotype using the clock gene expression oscillations. And the current methods are subjective, invasive, impractical, and can be inaccurate, and using blood collection, smartwatches, or self-reported questionnaires. So I'm using hair follicle cells and determining a more practical method. As current research has found that hair follicle cells are able to express these clock genes, I would like to see if we could see these oscillations with the gene expressions. To do so, I collect 10 hair follicle samples from 2 female and 2 male volunteers every 4 hours in a 24-hour cycle, so 9 a.m. to 9 a.m. I then place these hair follicle cells immediately into a freeze-thaw cycle that just opens up the cells for RNA isolation. I then run the RNA through a 2-step RT-qPCR reaction, which is basically just a reverse transcription reaction to produce cDNA into a quantitative polymerase chain reaction to amplify the clock genes of interest and see their expression levels over 24 hours. I then normalize this data using a housekeeping gene. This is just a gene that is expected to be expressed in all samples. So first I want to show you a very, very simplified schematic of the circadian rhythm and the regulatory feedback loop that controls it. The core loop starts with the clock and BMAL complex that activates the period and cryptochrome negative regulator complex. The period and cryptochrome then inhibits the expression of BMAL and clock, thus inhibiting itself and allowing a relief of BMAL and clock and starting a new cycle. At the same time, the genes reverb alpha represses BMAL expression, and the genes of ROR activate it. So after understanding that feedback loop of the BMAL and period genes, we can expect that these genes will be in antiphase as they're inhibiting and activating each other. So this is the graph showing the female and male volunteers and their gene expression for BMAL over a 24-hour cycle. You can see that the peak is at 9 p.m. and the trough is experienced around 9 a.m. for both female and male. And when looking at the period 1 gene, we see that the peak is at 9 a.m. and the trough is at 9 p.m. So antiphase as expected, and this is also seen with period 3. So I also ran this on 5 other genes, but for the interest of time and clutter, I summarized everything into a gene phase distribution graph showing the male, female, and then all volunteers together. And the gene phase distribution graph basically just shows the peak gene expression levels for the circadian clock genes. So we can see here that they are somewhat grouped together. They're somewhat grouped together and they're not randomly expressed. Otherwise, the points would be dispersed throughout the graph. So, for example, BMAL, all volunteers have a phase distribution around 9 p.m. and for period 2, 3, and cryo 2, it's around 9 a.m., so antiphase again. This just shows that hair follicle cells are able to express a robust circadian clock, providing a non-invasive and practical sample option.
For continued research, I would like to run this on more normal volunteers along with chronically ill patients, determine if there's a chronotype difference between the two. I'll also like to use the program Cyclops, which can decrease the number of time points to actually just one time point and predict the rest of the oscillations for 24 hours. Overall, this project provides a means of assessing chronotype that optimizes both patient comfort and data accuracy, and it allows accessibility to assessing oscillations of circadian control genes to be used to determine optimal times for drug effectiveness and chronopharmaceuticals. Thank you. Any questions?
Thank you. We have a question there, please.
How many chronotypes are there?
Well, typically there's the night owl and the early bird or early morning bird, but it's really just based on your circadian rhythms, how you behave, how you sleep and wake, and but typically it's those two.
One more question. Any other questions?
Yeah. Can you describe how did...
Wait, I can't find you. Oh.
Can you describe how the setup for volunteers worked? Like, did you have them like on site for 28 hours, and like, were they sleeping like, or can you describe the setup?
So I can't say their names, but thank you to them because they're in my lab. And basically we were just on site sleeping for 24 hours, and we woke up every 4 hours, so 1 a.m., 5 a.m., 9 a.m., etc. It was like a big sleepover. So. Yeah.
Any other questions? Were there pajamas at the sleepover?
There were.
Awesome. Thank you.
At least I had them. But.
No promiscuous bowls.
No.
Thank you so much. Thank you, Chloe. All right, here's the deal. We're going to take a stretch for just a few moments, but you don't get to leave because you won't come back. So please stand up. Let's stretch together a little bit. We've heard 8 amazing presentations so far this morning. So thank you all. And let's take a quick stretch, some deep breathing.
So if y'all want, we could actually do an exercise. Just reach up towards those stars that so many of y'all are studying, those black holes. Reach up, up, up. And then for those of you who work in labs, you have to reach down into that bottom cabinet for that reagent somebody forgot that may or may not still be in date and legal. Okay, just move around a little bit. And again, we're not going to leave. We're going to sit right back down and go ahead and get started with our second half. Thank you all so much.
I love the stretches. All right, about 1 more minute to get yourself resettled, please. All right. We'll have lots more time after this is over to connect. So, see, we kept everybody here and stretched out. Good, thank you so much. So, we have another 8 wonderful presentations coming up in this session. So let's begin by welcoming McKenna Taylor, and she's going to talk about some genetic variations and quorum sensing. Thank you.
Hello, so my name is McKenna Taylor, and I'm currently a senior at Florida Tech pursuing a double major in astrophysics and astrobiology. For the last couple of years, my research has focused on quorum sensing, which is a type of cell communication that couples cell density with phenotype or physical change. This was originally discovered in bacterial cells, but was later found in fungal eukaryotic cells. A few years ago, the PhD student that I do research for was able to discover a potential quorum sensing response in a non-fungal eukaryotic cell. And this was in the algae Chlamydomonas reinhardtii. She found that the strain of Chlamydomonas run24 caused an increase in swimming speed as it reached what we consider to be a high cell density threshold. We have since been studying the different variations of this type of this type of algae to see if other strains will also produce some type of quorum sensing response, to see if this might be something that we need to look for in other types of non-fungal eukaryotic cells, or if this might be just a one-off occurrence. For the last year, we've been studying the strains 408 and 3269, both of which were collected or produced by the scientist Ruth Sager. 3269 is actually a cross between a group 21 strain and the 124 strain, where this increase in swimming speed was originally discovered. For this process, we've been taking these strains and growing cultures on media plates, transferring them to a flask of tap media, which is tris acetate phosphorus or phosphate, and watching them as they grow from a low salinity to a high salinity threshold. At each stage, we have recorded that growth rate and taken videos to monitor that type of swimming speed. Once we have these videos, we're able to take a high salinity extract and add that to the low salinity culture to see that also elicits a quorum sensing response. Using these videos, we use the programming Fiji and ImageJ to track each individual cell and to track their average, minimum, and maximum swimming speeds. Once we have this data, we're able to create graphs showing what this change in swimming speed is and if there is a change from this low salinity to the high salinity. From this first graph, you can see that we did have a little bit of trouble figuring out how to get these cells to grow. The strain 124 is very commonly found in labs, as it is known for being very easy to grow and maintain. So this might just be a much easier type of strain to grow, and that's why we could have seen a much stronger growth in that rate. But there also could have been issues with contamination, which we will also be focusing on in the future when we come to future experiments. From these other two graphs, you can see that both of these strains did elicit some type of quorum sensing response. For the 3269, this did indicate a speed up in the swimming speed, which we expected as this is a cross between that 124 strain. So you can see that there is a much higher number of cells moving at a higher speed once we reach that high salinity, and a higher number of cells moving at a lower speed at the low salinity. But the 408 actually elicited a different type of quorum sensing response. It tended to slow down as a result of high salinity rather than speed up. So there's a much higher number of cells moving at a higher speed at a low salinity, and a higher number of cells moving at a lower speed at a high salinity. So because of this, we do think that these strains both elicit this type of quorum sensing response, and we can study them to determine which molecule is causing this quorum sensing, but they're different responses than we would typically expect from that 124. For the future, we plan to add nitrogen to our media to see if that lack of growth was caused by a lack of nutrients, and to see if this increased nitrogen would allow for these cells to grow a little bit faster at the rate that we would like to see. We will also be adding antifungal media plates to our experiment to see if that can potentially reduce any contamination that might have occurred. We will be testing more strains of this Chlamydomonas reinhardtii to see if light tendency or temperature tendency will have any indication on this quorum sensing, and we will be testing each of these 408 and 3269 cultures, the low salinity cultures, with the high salinity extract from the 124 to see if that also elicits some type of quorum sensing response. And I would like to thank Dr. Palmer's research lab at Florida Tech and all of you for allowing me to present today.
Thank you, McKenna. Do we have a question? Any questions? Aha. No questions? McKenna, thank you so much. Next, let's welcome Sophie Keel. She's going to be speaking to us about the efficacy of inactivated lactic acid bacteria vaccines.
Hello, everyone. My name is Sophie Keel, and I study microbiology at Colorado State University, and I'm going to be talking about the efficacy of inactivated lactic acid bacteria vaccines. So first, I want to describe what I mean when I say lactic acid bacteria vaccines. We begin with the lactic acid bacteria, and we use this because of its natural benefits, including it's a probiotic, it's naturally good for your microbiome, and it's already found in your gut innately. It also is good for when we're targeting mucosa, so it's delivered orally, and then it's good for viruses that are also trying to target your mucosa. It's easily produced and stored. It doesn't have a cold chain requirement, so it's easily distributed to warm countries, especially with a lower health income. It's also easily modified, so we have developed constructs against rotavirus, West Nile virus, and SARS-CoV-2. And we begin with our lactic acid bacteria, and the way we develop it into a vaccine is by adding two components. The first is a viral peptide, so a small subunit of a protein. We integrate it into the genome of our bacteria, and then it is expressed on the surface so that the immune system can recognize this small piece of a virus. And then the second thing we add are adjuvants, which just help alert the immune system to the presence of our vaccine. It helps them adhere to immune cells and induce immune-stimulating pathways. A question we have, however, is given that these are lactic acid bacteria, they secrete a lot of lactic acid as part of their metabolism. But lactic acid, we found in some literature reviews, can reduce immune responses. It inhibits immune pathways, reduces pro-inflammatory cytokines. And usually this is good. We don't want our gut reacting to everything that we eat. But in the context of a vaccine, it's not as ideal. So I am coming in to answer our question of whether our metabolism of our bacteria is affecting the efficacy of our vaccine. And we're studying this by eliminating this lactic acid production. We do this using Mirasol pathogen reduction technology systems, which uses a combination of UV light and riboflavin to target and degrade DNA and RNA, so all our nucleic acids. And critically, it preserves the cell structure so that we maintain our viral peptides and our adjuvant expression. We want to look at a couple of things in our mouse models that we are using as our live organisms. The first is if we administer this vaccine, do we see a difference in lactic acid concentration in our tissues of interest? So do we see this in lymph nodes? Do we see it in Peyer's patches along the small intestines? And is this enough to register a difference in the cell level? Next, we want to look at T cell activation. So are we getting a specific immune response to our vaccine? And then finally, we want to look at antibody production, and that's critical for establishing long-term immunity as well. So we'll be looking at various isotypes of antibodies over time. I'd like to thank everyone in the Dean Vilander lab who helped establish this project, as well as the Goodrich lab for giving us access to the Mirasol pathogen reduction technology system, and of course, Astronaut family, as well as my actual family. So thank you.
Thank you so much. Who has some questions on lactic acid bacteria?
I can ask one.
Ask a question. I can ask a quick question. Whenever you have the vaccine, the lactic acid vaccine, are you giving it like intramuscularly?
No, it's orally delivered.
Okay, so it's orally delivered. Whenever they don't produce lactic acid anymore, does that affect the microbiome at all?
That we're not sure about because we have done microbiome studies with a control and then our lactic acid. We haven't done that yet with our inactivated bacteria. I imagine it would alter it quite a bit because any administration of a probiotic does dramatically affect your microbiome. Usually it resolves, it's like short term, it's like within a couple days, but I imagine it would have a large impact.
Okay, gotcha. Thank you. I actually have one more question. Can I
Okay,
ask
yeah.
another one? Okay. So real quick, can you actually add more than one viral peptide to the bacteria?
We have not. I imagine you could. So right now we're using a small subunit of rotavirus for our main construct called VP8, but we also have constructs for VP7. I don't see why we couldn't do both in one, like a 2-for-1. I like it.
Cool. Thanks.
Thank you so much. All right, next let's welcome Diane Fakenled, and she's going to talk about the role of neuroinflammation in a couple of different ways. Welcome, Diane.
Good morning, everyone. My name is Diane Fakenlede. I am a senior biology major at Howard University, and I had the pleasure of working at the University of Wisconsin-Madison in studying the role of neuroinflammation in AAV human tau rat model of tauopathy. That's a lot of words, but first I want to talk about Alzheimer's disease. As we all know, Alzheimer's disease is a neurodegenerative disease that's associated with neural death and memory loss. But first, I want to show a quick side-by-side of a normal brain and the Alzheimer's disease brain. As we can see, the Alzheimer's disease brain is significantly lower in brain volume, and this is due to nerve death. And Alzheimer's disease, unfortunately, affects 6 million people in the United States, making it the 7th leading cause of death in the United States. And studies have found that Alzheimer's disease is caused by beta amyloid plaques and neurofibrillary tangles. Neurofibrillary tangles are caused by tau aggregation. But what is tau? Tau is a protein that binds to microtubules in the cell. These microtubules assist in cell assembly for these nerve cells. And when the tau protein is activated at high amounts, or also known as hyperphosphorylation, it could cause them to clump together or aggregate. And studies have shown that in Alzheimer's disease, cognitive decline does not occur until neurofibrillary tangles are shown. So this leads to the preliminary project. First, we wanted to see if the expression of human tau in the rat hippocampus result in the hyperphosphorylation of the tau protein. And also, it will be interesting to see if there's neural loss associated with the expression of human tau. And we believe that the expression of human tau results in a pathology that resembles human tauopathy, also known or such as Alzheimer's disease. But how are we able to express human tau in the rat brain? We use a virus, an adeno-associated virus, also known as AAV. So we are able to develop a human tau vector that is injected in the hippocampus. We are interested in the hippocampus because it's the region of the brain that's the most vulnerable to Alzheimer's disease. So we're able to inject this vector, and over time, the human tau protein is able to be expressed in the region of, in the target region. And we're able to make sections of this. We wanted to test multiple groups. One group was uninjected, another group was injected with green fluorescence protein that served as our technical control, and the group that is injected with the human tau protein. And these injections occurred at different time points, 3 weeks, 8 weeks, and 12 weeks. And this summer, I was responsible for 6 months, but unfortunately, we didn't have the time to take pictures when I was there. But I was able to conduct histology on them. I conducted initial staining, which stains for viable and non-viable cells, and also an immunohistochemistry, which we use antibodies against hyperphosphorylated tau and human tau. So these are the preliminary results. As you can see, the brown that we see here represents expression. So here, this is the expression of tau that has been activated at abnormal amounts. And in all 3 time points, we can see that there is hyperphosphorylated tau in the rat cohort that has been injected with human tau. And in human tau, you can also see expression there in all 3 time points. And also we looked at viable and non-viable cells, and you can see over time the hippocampal layer decreases, and this is the hippocampus right there. So to conclude, we can say that there is robust expression of the human tau and hyperphosphorylated tau in the rat hippocampus, and also an increase of neuronal death in the rat hippocampus. And we are doing this project to be able to use the rat model as a good model to look at Alzheimer's disease and human tauopathy. And in the future, it will be interesting to see how cognition is affected due to the expression of human tau, because as I stated in the beginning, cognition does not decline until we see neurofibrillary tangles. So that will be interesting to see. And I would like to give my acknowledgments to my PI, Dr. Karina Berger, my scholarship program, the Carstone Scholars program, and also the ASF family for allowing me to present here. And yeah, I will take any questions.
Thank you so much. There's a question?
Yes.
Hi, quick question for, unfortunately, a family that has suffered from Alzheimer's. Do you see your research, or would the goal ultimately be to better detection of Alzheimer's and obviously most importantly prevention?
Yes, thank you so much for your question and for being vulnerable. I do believe that this research is very key in the advancement of the prevention of Alzheimer's disease. Right now, there are treatment plans to remove the plaques, but we have seen that with the removal of the plaques, there is still cognitive decline. That's why we're looking at tau. And hopefully, in the near future, we're able to use this research to be able to prevent cognitive decline over time.
Good. One more question? Go ahead.
Prior to like perfusion in the histology assays, did you do any behavioral assays to see how this affected the rat's behavior and
Yes.
memory and everything?
Yes, the lab did conduct some behavioral exams. It's a small cohort to really see like any changes in like the cognition. However, we will conduct more exams on that. Thank you.
Thank you. Such important research on a very difficult disease. Thank you so much.
Thank you.
All right, next let's welcome Neha Skandon, and we're going to talk about receptor antagonism. Let's go with just receptor antagonism. Welcome, Neha.
Good morning, everyone. My name is Neha Skandon, and I'm a student at the Johns Hopkins University, and I conduct research at the National Institutes of Health. And so today I will share some of my work on the neurobiology of addiction, focusing on opioid use and alcohol use, specifically glucocorticoid receptor antagonism. So to provide some brief context, so the opioid epidemic in the United States has been increasing exponentially over the past 2 decades. In just 2023 alone, there have been more than 100,000 overdose deaths. And did you know that makes overdose the leading injury-related cause of death in the United States? And so you may have seen the recent series of articles published in the New York Times focusing on the state of this epidemic. But what is not as frequently discussed, but remains a very interesting area, is what and how does substance use work in the brain? So what exactly is that neurobiology that underlines these cases? And so what I hope to do today is to shed some light on that biological perspective for you through my work. And so imagine opioid use disorder and alcohol use disorder as a feedback loop in the brain. And so essentially substances act as stressors that activate the HPA axis and also their associated receptors, and that includes the glucocorticoid receptors. And that forms the central stress feedback system in the brain. And so when substances are unavailable after prolonged use, then that activates these withdrawal symptoms, a multitude of them. And one prominent symptom is enhanced pain sensitivity or hyperalgesia, which is the focus of my research. And essentially what is thought is that these symptoms continue to drive that continued substance use. And so given that, is there a way that we could potentially mitigate some of those symptoms to try to block that continued reinforced substance use? And so my objective is to determine the effect of pharmacologically blocking glucocorticoid receptor antagonism on the presentation of hyperalgesia. And to do so, I utilized a preclinical animal model utilizing rodents. And so to briefly touch on methods, rats were either administered with heroin or alcohol, and so they were dependent to heroin or alcohol, and then they were treated with either mifepristone or miricorilant, which are 2 GR antagonists, so essentially they block the glucocorticoid receptors in the brain. And so once we administered them with the substances, then I proceeded to test them for hypersensitivity to pain as they underwent withdrawal. And so some key findings, we first observed that mifepristone seemed to decrease thermal hyperalgesia in heroin-dependent male rats, and that miracorilant acted in a similar manner in female rats, in that they decreased mechanical sensitivity to pain. What we also saw is that mifepristone mitigated alcohol withdrawal-induced hyperalgesia in male rats, and this acted in a dose-dependent response. So higher doses of mifepristone seemed to reverse hyperalgesia, which was induced in alcohol-dependent male rats. And so these findings suggest that glucocorticoid receptors in the HPA axis are a potential therapeutic in treating pain symptomology during withdrawal. And given that withdrawal symptomology seems to continue to drive this reinforced substance use, at both the preclinical and the clinical levels of research in this field, it's becoming increasingly important to see if we can address withdrawal symptomology to improve treatment for substance use disorders. And so I would like to thank my PIs, my mentors, Dr. Vendrusco and Dr. McGinn, who have supported my research over at the NIH. And I'd also like to thank my PIs at the Johns Hopkins School of Medicine, Dr. Hune, Dr. Ellis, and also Dr. DeMarlo, who I work with throughout the year, for all of their mentorship and support. And I also thank the Astronaut Scholarship Foundation for this incredible opportunity and for allowing me to present today. So thank you, and I'm happy to take any questions.
Thank you so much. Oh, it's a race. There we go.
I have a question back here. Hi. I was curious what your decision was to specifically look at opioid addiction and alcohol addiction, and if this research would be prevalent for other addictions.
Absolutely. And so my lab particularly focuses on alcohol and opioids, and the reason for that is because of the opioid epidemic, and so that has particularly made opioids a key interest in drug addiction. In addition, alcohol also remains the #1 substance that has been overused, and so because of that, that's also a key substance of interest. But given that substance use acts in a similar manner, like neurobiologically, these models could definitely be translated to other substances as well.
One more question.
I had a question about why did you use different time points for the male and female rats?
Absolutely. And so this was found in previous studies that female rats entered withdrawal in a sooner period than male rats. And so because of that, I tested female rats at a sooner time point.
Thank you. Such another important topic to our world. Thank you, Neha.
All right, thank you.
Next, let's welcome Bryce Wilson, and he'll be speaking to us about temporal coordination of the cellular response to oxidative stress. I'd like to know how to respond to stress. Let me know. Thank you.
Cool. So my name is Bryce. I'm a senior at the University of Arizona, and I'll be talking about the temporal coordination of the cellular response to oxidative stress. So in my lab, we're interested in answering the question, how do cells respond to stress? Not necessarily the type of stress if you're feeling from schoolwork. These are more cellular levels of stress. And we look at this through examining transcription factor dynamics. So transcription factors are proteins that change the expression of various genes in response to different stimuli. In this diagram, you can see two transcription factors, FOXO and P53. If you're in the cancer world, you might be familiar with P53. And they respond to a wide array of different signals and elicit or activate various outcome pathways. Both FOXO and P53 respond to oxidative stress. But what is oxidative stress? Reactive oxygen species are byproducts of normal metabolism and typically produced within the mitochondria of the cell. And hydrogen peroxide is one of these really important cellular ROS. And interestingly, oxidative stress has been implicated in various cancers, neurodegenerative disease, as well as diabetes and cardiovascular disease. And hydrogen peroxide at low levels is actually really good for cells. It signals growth and division, and they need it to survive. But at really high levels, this is what we call oxidative stress. It's quite toxic and ultimately leads to cell death. To examine how cells are actually responding to oxidative stress, my lab looks at transcription factor activation over time. So we use live cell time-lapse microscopy of cells treated with hydrogen peroxide, and we do this mostly using CRISPR to endogenously tag proteins with fluorescent proteins, so you can visualize these just with standard epifluorescence microscopy. Now I'm going to show you a video on the next slide, and I want you to pay attention to the cell that the arrow is pointing to, and these graphs at the bottom are essentially just quantifications of hundreds of cells of this same type of video. And at the 0 time, so the arrow is pointing to the same single cell, just the left panel is FOXO and the right panel is P53, and this is over the same time scale. At the baseline time 0, there's neither protein active in the nucleus. At 2 hours, you see FOXO is active, FOXO goes away, and then you see P53. And then for the most part, most of these cells don't have both of these proteins active at the same time. So you see this burst of FOXO and then P53. But it's actually not just FOXO and P53. This is a very widely coordinated response. So when we looked at other transcription factors, we found that there were several activated with either FOXO or P53, but not both. And this led us to ask the question, how are cells coordinating this activation over time? And we found the answer in this really cool enzyme family called the peroxyroxins. The peroxyroxins are antioxidant enzymes that scavenge hydrogen peroxide and protect cells from oxidative stress. Interestingly, several RNA-seq transcriptomic studies have found altered expression of the peroxyroxins in various cancers, as well as neurodegenerative disease, particularly in Parkinson's. What's really cool about these proteins is they have these reactive cysteine residues that serve their catalytic function and reduce hydrogen peroxide, but also these residues have been shown to directly form disulfide bonds with target proteins and activate signaling pathways in that manner. And this signaling function is actually how this relates back to the transcription factor activation. So we demonstrated that the specific oxidation state of those cysteine residues in the peroxyroxins is what's coordinating this response over time. And if we artificially modulate that oxidation state, we can shift the temporality of this response. And just some kind of takeaways and implications. If you took away anything from this short talk, I hope you understand now that oxidative stress is a really, really important disease process, kind of in a wide range of diseases. Also, single-cell quantification of live cell imaging can tell us really a lot of information about cells and protein dynamics, and some implications largely with targeted therapeutic design. So the peroxyroxins, there's actually a small molecule inhibitor that can modulate the oxidation state, and this molecule called J14 has been shown in vitro to preferentially kill cancer cells. And thank you to my lab members, a wonderful graduate student that I worked with, Dr. Jose, funding from the Beckman Foundation, as well as an R01. And all this work and much more was published in Nature Communications back in April and available at that QR code.
Thank you so much. Who has a question for Bryce? Come on, killing cancer cells? Somebody's got to have a question. No? You did great. Thank you so much. Oh, wait, there is one. Yay. Late breaking news.
So what was the main method that you were inducing oxidative stress in your samples?
Yeah, so we actually just, I probably should have mentioned this. So all this work was done in MCF7 breast cancer cells and then validated in other cell lines, but we literally just add hydrogen peroxide to cell culture media. So it's exogenous application. There are some systems to induce endogenous hydrogen peroxide action within a cell, and you can actually localize. It's through an enzyme called D-amino acid oxidase, so it takes the D isoforms of amino acids and produces hydrogen peroxide, and you can localize it to different places in the cell as well. Yeah.
Thank you so much, Bryce. All right, next let's welcome Devarshi Mukherjee, and we're going to be talking about investigating the role of osteopontin/secreted phosphoprotein. Woohoo!
Hi everyone, my name is Devarshi, and I'm from the University of Michigan, and I will be talking about the investigation of SPP1 in the radioresistance of H3.3 G34R gliomas. So just to give some background, what is glioma? Glioma is one of the most common forms of brain cancer. About 33% of all brain tumors are gliomas. And currently, we have quite the dire need for improvements in treatment. The 10-year overall survival for patients with glioma is still less than 1%. So the tumors that I specifically study are called H3.3 G34R gliomas. And if you guys remember from Shelby and Lily's presentations, we covered a little bit about chromatin and histone proteins. All this is referring to is just a mutation in histone 3.3 that causes some epigenetic and genetic hallmark characteristics of these tumors. And I will mention that the G34R gliomas are most commonly found in the pediatric population. So in the context of G34R gliomas, I'm specifically looking at radioresistance. Many of you may be familiar with radiation therapy as a very common treatment method for basically every form of cancer. And so I'm specifically studying radioresistance to potentially increase the efficacy of radioresistance, eliminate resistant cell populations, and ultimately try and avoid having resistant cell populations that almost inevitably lead to tumor recurrence in basically all patients with glioma. And my project kind of started after we just observed a phenomenon in our lab's mouse model of H3.3 G34R gliomas. And that was, you can see here that in resistant mice, during treatment, the tumors will decrease in size. However, after, they become very aggressive and eventually kill the mouse. Whereas radiosensitive tumors, the treatment basically eradicates the tumors completely, and even with re-challenge of the tumor cells, they do not develop the cancer. So essentially, we want to figure out why this is happening. The method that we are using is single-cell RNA sequencing. For those of you who aren't familiar with single-cell, essentially you get a transcriptomic picture of gene expression across tens of thousands of genes for every single cell in the tissue samples that you submit. So if you see over here, this is just a visual depiction of the data with some labels, and each and every dot is a single cell in the sample, and they're kind of graphed according to about 30,000 dimensions, which are compiled down into 2 dimensions that you see here. And then I've run a variety of analysis techniques on this to try and investigate this problem. And after a lot of analysis and corroboration with the current literature, I have identified a molecule known as secreted phosphoprotein 1 as the most biologically relevant target. And essentially, so this is ligand receptor analysis. If you look on the right side, you'll see here, this is just blown up right here, but SPP1 is extremely highly secreted in radiosensitive mice, whereas in the radioresistance mice, you can see that there's almost none. So looking further into that ligand receptor analysis, here are the interactions between the tumor cells and the tumor microenvironment, and this R is resistant, this S is sensitive. And you can see in the resistant tumors, there's 0 expression of SPP1 from microglia, which are a type of macrophage in the brain. And in sensitive samples, there's a lot of activity. So essentially, what I'm currently working on is investigation of this result in vitro and in vivo. I'm currently co-culturing microglia and tumor cells and trying to investigate whether they have increased secretion of SPP1 in the sensitive samples, and also looking at other cytokines such as CCL5, which you can see here, which had some interesting results, which I can talk about later. And then finally, I'm also conducting my own in vivo experiments where I'm doing serum measurements of SPP1 from the blood in sensitive versus radioresistant mice, and I'm also doing immunofluorescence studies of the brains to look for SPP1 presence. And overall, the goals and implications of this are to understand, kind of add to this really complex interactions between microglia and tumor cells, as well as the tumor microenvironment, and hopefully evaluate whether SPP1 is a potential therapeutic target for these tumors that maybe one day we can go to clinical trials. So with that, I want to thank my PIs, my lab mentors, all of our collaborators, funding sources, and most of all, ASF for the opportunity.
Thank you so much. we have a question. Good.
Fascinating research. Just out of curiosity, why is this particular type of cancer more prevalent in pediatric patients as opposed to adult?
So that's a very interesting question, a good question. It just so happens that the people who end up with this mutation, like genetically, the tumors arise early in life, whereas other tumors, for example, you may know people who have brain tumors, those mutations are acquired mutations, but this is inherited. Not inherited, sorry, but you're born with it.
Another question?
I
had a question.
Oh, go for it.
I was wondering if you know if this gene SPP1 is only involved in like the one type of cancer you're looking at, or is it across all different types of cancer? And then also, do you know if it's not only related to radiation resistance, but also like chemotherapy resistance or other types of treatment?
Very good question. So yes, it has been implicated in some other cancers, I believe thyroid cancer, but as you may know, the way molecules work in different contexts can be completely different. So there's very little research about radioresistance with SPP1 in glioma. However, there's actually a lot of conflicting evidence. That's why it's a bit of a murky field, and hopefully we can elucidate something through my research.
I'm going to ask an online question.
Okay, cool.
So we get some representation. So the question was, are the cancer cells really hard for you to culture, or how easy is that?
Actually, the ones I'm working with right now are actually pleasantly easy to work with. They grow very fast, which obviously is not good for patients, but is good for research. And yeah, they're pretty resilient, so yeah, good question.
I'm so happy to hear the cancer cells are cooperating with you.
Yeah, unfortunately.
Unfortunately. Thank you, Devarshi. Very good. Wonderful.
Thank you so much.
Thank you. Now we would like to welcome Amanda Galloway. She's going to talk with us about characterizing triple-negative breast cancer metabolism upon LDH-B inhibition. Welcome, Amanda.
Hi, everyone. My name is Amanda Galloway. I am a senior biomedical engineering student at Texas A&M University, and I'm also going to be talking about cancer. So I'm going to be talking about in vitro metabolic analysis of LDH-B inhibition in triple-negative breast cancer. So first, to start off, I'm going to talk about metabolic symbiosis in tumors, which is primarily responsible for how tumor cells proliferate so fast. And this is definitely seen in triple-negative breast cancer, which is a pretty aggressive form of cancer. And so what happens in metabolic symbiosis is that there are glycolytic and oxidative tumor cells within the tumor microenvironment, and they actually work together by lactate dehydrogenase B, which is the enzyme that converts lactate back into pyruvate. And so excess lactate from glycolytic tumor cells are transferred back into pyruvate in oxidative cancer cells, and this speeds up metabolism, which is problematic for cancer. So theoretically, if we can inhibit LDH-B, we can possibly slow cancer growth. And so in 2021, the first uncompetitive selective LDH-B inhibitor was discovered, and so theoretically, if we can use this inhibitor, we can study cancer metabolism and how metabolic symbiosis works a little more by disrupting it. So how do we do this? So I use fluorescence lifetime imaging microscopy, otherwise known as FLIM, which is a huge area of research within my lab, and fluorescence lifetime imaging microscopy is a non-invasive imaging modality. And so in figure 1, you can see the multiphoton microscope diagram that we use. So we use a 2-photon system. And then in figure 2, you can see the fluorescence excitation and emission spectrum for NADH and FAD, which are 2 metabolic coenzymes that have separate fluorescent lifetimes. So it allows us to image them and get some information out of them. And then getting the fluorescence intensities from NADH and FAD, we can create what's known as the redox ratio, which is seen in equation 1. And the redox ratio is a direct measurement of metabolism. So the experiment that I designed is to take MBA MB231 cells, which is a triple negative breast cancer cell line from MD Anderson Cancer Center patients, and we plate them in imaging dishes and allow them to grow for 24 hours. And then after that, we will add AXKO0046, which is the LDHB inhibitor, into the imaging dishes and allow that to grow for another 24 hours. And then after that, we complete fluorescence lifetime imaging experiments using our multiphoton microscope. And so some preliminary imaging results that we have gotten. So over on the left, you can see some of the redox ratio sample images. So the scale on the right shows the redox ratio that correlates to the color. And so in image A, there is no inhibitor, so those are healthy cancer cells. And then image B has 0.1 of the micromolar of the inhibitor, image C has 1 micromolar, and image D has 10 micromolar. And so we can see that with the increasing inhibitor concentration, that there is a morphological change within the cells, and their color is changing a little bit, meaning that the redox ratio is changing. And also, it's not very representative in these pictures, but when we're looking at the whole cell, the confluency is also changing, meaning that some of the cancer cells are dying. And so some more quantitative results seen in the box plot in figure 4, that's just the redox ratio of the same cell groups at varying concentrations. And we can see that there is a steady decrease as the concentration of the inhibitor increases, meaning that metabolic symbiosis is being disrupted and that the cells are less able to metabolize in the way they want to and the way that benefits them. So where is this heading? So in the future, I want to do more cell viability assays just to get a better idea of how quickly the cell populations are changing upon LDHB inhibition. I also want to repeat the imaging experiments I've already done, but at 1 hour and 48-hour time points to just get a better idea of how quickly this happens. And then lastly, I want to perform all of these experiments on MCF10A cells, which is a healthy epithelial mammary cell line, because theoretically, if we can do all of this LDHB inhibition on healthy cells and not have an effect, then this could be a treatment option for triple negative breast cancer that only selects cancer cells. So with that, I would like to thank my quantitative optical imaging lab at Texas A&M and all of you today. Thank you.
Thank you. We have a question in the blue shirt right there. Right, I saw back there.
Okay.
No?
Hi. So I'm wondering if you mentioned the metabolic equilibrium between glycolytic and oxidative pathways in triple negative breast cancer. Is that only specific to triple negative breast cancer, or is it also present in other types of cancer cells? So for example, could this therapeutic approach also be applied to other types of triple negative or other types of breast cancer that maybe develop resistance to conventional therapeutics?
Yeah, so the reason I'm looking at triple negative breast cancer is because it's typically the hardest to treat, because other breast cancers usually have a receptor or hormones that other treatments are pretty effective for, but in triple negative breast cancer, all of those are lacking, so there's nothing for the treatment to attach to. So that's kind of why I'm looking at that interaction between the glycolytic and oxidative cancer cells. And metabolic symbiosis has been found in other cancers for sure, but I'm just not sure how changing that reaction in other cancers would work, but certainly maybe some of the more aggressive tumors that aren't responding to other traditional therapies. So,
one more question.
So microscopy is really cool, but also on the images, why do you think as the concentration goes up, you almost see a change from these really small, lots of little redox foci into these really large pockets?
Yeah, so I think that is definitely just because, so as the concentrations are increasing, we can see a change in the shape, and that change in the shape is indicative that they're no longer healthy. So in image A, we can see healthy cancer cells, and then in image D, they're much more round, and you can see those pockets of the redox ratio. So changing, and so what I think is happening is I think they are dying, and so that's why you see those darker colors there. I think the cells are dying. And so that's why I want to do it at 48-hour time points, because I wonder if I would even be able to see anything at that point, or if they would all be dead, or just kind of what that looks like. So
Thank you so much, Amanda. Okay, next we'd like to welcome Audrey Rips-Goodwin, and she's going to talk to us about correlating lateral hypothalamic neuronal activity. Welcome.
Alrighty. I am Audrey Rips-Goodwin. I attend the University of Kansas, but today I'm going to talk about my research that I did this summer at the National Institute on Drug Abuse, where I used 2 machine learning pipelines, SLEEP and SIMBA, to analyze animal behavior so that I could correlate that with the neuronal activity of these LH-VGAT neurons. So, what are LH-GABAergic neurons? The lateral hypothalamus, or the LH or LHA, is a subcortical brain region that's really small but really powerful. It's known to be involved with motivated behavior, homeostatic regulation, as well as feeding behaviors, which is what we were interested in. Now, there are 2 main subpopulations of these LH neurons. There's the GABAergic and the glutamatergic neurons. We are focused on those GABAergic neurons, and GABA is an inhibitory neurotransmitter, and so GABAergic just means that they express the receptor for that GABA to bind to, and so we were looking at those that express the vesicular GABA transporter. And LH-VGAT neurons as a whole are involved with food seeking, consumption, and positive energy balance, so making sure that your body is getting enough food or that these mice are getting enough food. And it is hypothesized that 2 subpopulations of these neurons uniquely encode for appetitive and consummatory behaviors separately, so acquisition of food or engaging with sensory stimuli as opposed to actually grabbing and eating that food. And so this reflects how complex feeding and consummatory behaviors are. And as you can imagine, if we're trying to examine these behaviors in animals, it's really difficult because these behaviors are distinct yet oftentimes overlapping. And so that leads to this problem where researchers have had to, until recently, manually watch videos of mouse, so like watch mouse TV, and manually score these videos. And I had to do it too. It's pretty boring. And not only is this time consuming, but it's very error prone. Like imagine between individuals and also within an individual. And so the purpose of my project was to implement 2 machine learning pipelines that happen to be in Python to get animal pose estimation data, so track the coordinates of the animal across frames, as well as track and label their behavior. So I used 2 pipelines, SLEEP and SIMBA, to then extract the timestamps at which behavioral bouts are occurring and correlate that to the neuronal activity of these LH-VGAT neurons using in vivo functional calcium imaging data. So this is just kind of a workflow of what I did. We started by recording both the functional imaging data in the live mice as they're running around, being exposed to chow, as well as recording their behavior. So I began by labeling select frames, training a model, predicting on new frames, correcting those, and continuing this iterative process until the model achieved the accuracy that I wanted. And then I could batch predict on new videos for new animals and extract their coordinates of their body parts. Using those coordinates, as well as that raw video footage again, I used another pipeline, SIMBA, to extract features like movement, position, velocity between frames, and then I could go ahead and do some region of interest analysis and look at how much time the animals were spending like near the chow in the food zone. And this could be used for different behaviors. We were specifically just looking at feeding. And then we get to the big shebang. We can annotate behavior, train a machine model to label behavior for us that is more consistent than a human. And so with that, we can take the results of that model and extract the timestamps and coordinate that to the functional imaging and look at the specific consummatory behaviors and what's going on in the brain. And then we can get cool, pretty data like this. This is very preliminary data for 1 mouse in 1 video. And on the y-axis, we have the 12 neurons that we could see in this mouse in this 1 video. And on the x-axis, we have the time relative to the initiation of a behavioral bout. On the left-hand side, we have touching, so like just that sensory stimuli. And on the right hand, we have eating and then grabbing and eating, so the mouse goes like that. And then the shading indicates the neuronal activity normalized to baseline. So up here in yellow, we would have increased activation of those neurons, and down to blue would be decreased activation. And as you can see in this one example, there's not really much going on here
associated with touching. But when that mouse starts eating that pellet, which is not, it's just basic food. It's not super yummy to them or anything. But there is some increased activation here and some decreased activation, which is really exciting for us. And we, of course, need to further analyze this data, look at more mice and more videos. And we want to take this this was basically a proof of concept for the lab. I was just setting up these pipelines. And we want to look at more salient stimuli like high-fat diet or sucrose, which is kind of like not a full-on drug to the mice, but it's really palatable to them. And so with that, I'd like to thank my lab, the Aponte lab with my PI, Dr. Yeka Aponte at NIDA, as well as Akit Zelia Garcia, the postdoctoral fellow I worked with, the University of Kansas, where I have been for the last few years, as well as the Astronaut Scholarship Foundation and family for bringing me here today. I will now open up the floor to questions.
Thank you, Audrey. Who has a question?
Thank you so much for your research. So throughout the pipeline and the development of all of the computational systems that you did, which part was the most challenging for you?
These were tools that were free and open source out there to use. The hardest part was getting them installed and getting these GUIs, the graphical user interfaces, up and running. I spent 3 weeks doing that, just figuring it out. And then I got them up and running, and yeah. So that was probably the most difficult part. And getting the quality of, you need the video quality to be good so you can see the animal body parts throughout the whole video.
Any other questions? One more. There's one over here. Over there. Okay, go.
Thank you very much for that presentation. That was wonderful. So for the models, I assume basically you train the models in-house, so you basically set yes, no, one hard-encoded vector statuses toward you watch the video and say whether it eats or not, and then that's how you generate your data to pre-train your model, right? And then the second model associates that with neural activity, if I understood correctly.
So the two models, one is just tracking using neural networks to track the animal pose estimation between frames, so the coordinates. And then we feed that into another model that we can label the behavior, and then so using that data we can outside of a model coordinate it with neuronal activity from a functional calcium imaging or chemogenetics or optogenetics or anything that you want to do.
Thank you very much. And if I can quickly ask, how many videos did you need to use for training?
So that's a tricky thing. So I found out that I did things wrong at the end of the summer. But so you want to segregate your data, so you have your training data and then your testing data. So it kind of depends on what behaviors you're looking at, and it's really tricky because we don't have much extra data to work with. So it's so at least it just depends on how much data you have to begin with. But I think it was close to 60% of it was used for training. So in the future it'll be okay because we'll have models all ready to go to apply to new stuff. But in the beginning it's kind of tricky.
Okay, thank you so much.
Thank you.
So we have come to the end of the technical conference session number 1. We've heard about cancer, we've heard about addiction, we've heard about space flight, we've heard about many pieces of science that I can't pronounce. We've heard about Alzheimer's, and we've heard about what's in the water. Amazing. Give yourselves a huge round of applause. Amazing. So I want to thank all of the scholars who have presented. I do have your notes if you want to come up afterwards, and I want to encourage all of you to come back for this afternoon session because the opening act for that will be the rat cohort and the voles. So with that, thank you again, and we'll turn it back over to Carolyn.
All right, well, thank you, and thank you, Carol and Ashley, keeping us on track. Thank you, CCR, for AV working. We always appreciate that when we keep it smooth. when the clickers actually work, right? But Professional Expo is up next. We are wrapped up so that you have a nice little break. We'll keep it for the 11:30 start time. It might be able to open a little bit before that, maybe 11:15. Scholars and attendees, we will be starting promptly at 3 p.m. for the second portion of the Professional Expo today. So return here for part 2, just like any good trilogy. And with that, thank you so much. Another round of applause for the 16 presentations this morning.